Hello everyone. In the previous video, we discussed about Singer and Nicholson interpretation. In that, what they said, plasma membrane is made up of with proteins and phospholipids. Proteins and phospholipids, and it is trilamellar model. Trilamellar model. That means three layers are present. Three layers are present in the plasma membrane. Outer protein, next middle lipid bilayer and inner protein layer. These three layers are present in the structure of plasma membrane, so that it is called trilamellar model. And based on their interpretation and based on their views, let us draw the structure of plasma membrane. According to Singer and Nicholson, in the plasma membrane, outermost layer is protein layer. In the middle part of the plasma membrane, lipid bilayer is present. This is lipid bilayer. These are lipids, phospholipids. Okay. This is lipid bilayer. Glycerol heads and fatty acid chains are present. In this, fatty acid chains are facing each other. These are fatty acid chains. They are facing each other. And glycerol heads. These are glycerol heads. These glycerol heads are away from each other, and weak van der Waals forces hold these two lipid layers in the plasma membrane. You understand? Weak van der Waals forces present between these two layers, so that these two layers are held together. Okay. Next, what they said, outer protein layer is present. Proteins are generally alpha globular structures. These are protein layers. Okay. And these proteins are loosely arranged. Next, again in the proteins, two types are present. According to Singer and Nicholson, proteins are two types: extrinsic proteins and intrinsic proteins. Extrinsic proteins are present outside the phospholipids. They are present outside the phospholipids. These are extrinsic proteins. Next, coming to intrinsic proteins. Intrinsic proteins are partially or completely embedded in the lipid bilayer. Partially means like this. I am drawing here. This is a protein. This is partially embedded in the phospholipid bilayer. Next, coming to completely embedded protein. This is completely embedded protein in the phospholipid bilayer. I understand. Proteins are two types according to Singer and Nicholson. They are extrinsic proteins and intrinsic proteins. Extrinsic proteins are present outside the phospholipids, and intrinsic proteins are partially or completely embedded in the phospholipid bilayer. These are intrinsic, or otherwise they are also called partially embedded intrinsic proteins. They are completely embedded intrinsic proteins. Let us extend this structure outer to the next to the this completely embedded intrinsic proteins. Again, phospholipids are present. These are phospholipids. Okay, I understand. Outer to them, again extrinsic proteins are present. These are extrinsic proteins. This structure is explained by Singer and Nicholson. Singer and Nicholson also given a phrase to the structure of this plasma membrane. What is that phrase means? Protein icebergs. Protein icebergs in the sea of lipid. This given a phrase that is protein icebergs. Protein icebergs in the sea of lipids. This phrase is given by Singer and Nicholson for to understand the structure of plasma membrane. Here, in a sea, large sized icebergs are present. They are freely float in the water or fluid. Here, proteins are compared with icebergs. These intrinsic and extrinsic proteins are compared with icebergs, and C is compared with lipids. This lipid bilayer is compared with C, that is fluidic in nature, and in which proteins and lipids are moves from one place to another place. Based on that, this structure is named as fluid mosaic model. Fluidic. Here phospholipids are fluid in nature. They are semi-solid in nature. They are not completely solid and completely liquid. They are semi-solid in nature. In that, protein molecules are moving from one place to another place. Next, let us check the properties of plasma membrane. Properties of plasma membrane. What is the first property of plasma membrane? Plasma membrane allows the entry of water and fat soluble molecules. Okay. In that, for example, this is a fat molecule. Generally, fat molecules are allowed by proteins and phospholipids. Next, fatty acid chains, so that this phospholipid cross the plasma membrane. Next, coming to water. Entry of water is inhibited or hindered by fatty acid chains. Generally, these fatty acid chains will not allow the entry of water. This point is failed to explain the previous models, but here Singer and Nicholson successfully explained how the water entered from outer side to inner side of the cell through plasma membrane. Okay, how the water enters? Here, partially embedded intrinsic proteins are present. Again, completely embedded intrinsic proteins are also present. Along with this protein, here is water. This water enters through this protein and cross the plasma membrane and entered into the cell. This completely embedded intrinsic protein helps in the transport of water. That's why this protein is also called channel protein. You understand? This is called what? Channel protein. Sometimes they are also called porins. They helps in transportation of water which are present in the plasma membrane. They are called porins or channel proteins. Okay, first property is explained by Singer and Nicholson. Next point. Next property is protein phospholipid ratio is variable. In the plasma membrane, protein and phospholipid ratio is variable. That must be variable. That must be changed in the plasma membrane. Okay, let us check this point in the structure of fluid mosaic model. structure of plasma membrane according to fluid mosaic model. For example, here how many proteins are present? These are proteins 1, 2, 3, 4, 5, 6, 7, 8. 8 proteins are present. This is another one, 9. 9 proteins are present. Next, how many phospholipids are present? 1, 2, 3, 4, 5, 6. For example, 6. 6 phospholipids are present. How their number changes? Here, phospholipids and proteins. Phospholipids are generally moves in two ways. That is flip-flop movement and transition movement. First, coming to transition movement. In transition movement, what happens? This is one layer. Within the one layer, phospholipids moves from this place to this place. For example, this is A. For example, this is A, B, C. Here, C is moving from this place to this place, and A is moving from this place to this C place. Here, these two are moving from their positions to center. In this situation, what happens? Here, no phospholipid is present. Here also no phospholipid is present. Then the ratio will be changed. The ratio will be changed. There is no phospholipid. Their number is changing. Automatically, what happens? Their ratios also variable or changes. When the protein and phospholipid ratio is variable, automatically what happens? This plasma membrane becomes dynamic. Plasma membrane is dynamic in nature. Dynamic. Their proteins are floating in the lipid bilayer. They are freely moving from one place to another place, so that it is dynamic. It is flexible. This is about the structure of plasma membrane according to Singer and Nicholson. In the next video, I will come with another topic. Thank you.